A furious buyer logs onto a reptile forum. They've just spent top dollar on a highly sought-after amethyst purple lavender corn snake. But when they open the shipping box, the animal inside is a muddy, dull brown hatchling. They were promised vivid rose-pink saddles set against a shimmering silver-purple background. Instead, they're staring at an animal that looks like it was scooped out of a murky puddle. The confusion actually cuts both ways. Down the street, a novice breeder is looking at a clutch of eggs produced by two completely standard brown and orange wild-type corn snakes. To their absolute shock, one of those hatchlings emerges with distinct cool-toned lavender markings. To figure out why a premium purple snake arrives brown, and why ordinary parents suddenly produce a rare mutant, we have to look past the physical scales and investigate the invisible rules governing the animal's DNA. There are two specific mechanics at play here: the hidden genetic sequence that maps out the snake and the biological clock that ultimately colors it. The discrepancy between what was promised and what was delivered creates a mystery that can only be solved by examining how reptilian DNA and biological time actually intersect. The engine driving the lavender corn snake is a fully recessive mutation. This animated diagram illustrates how the three main reptile pigment channels work. In a wild-type snake, you have erythrin for red and orange, melanin for dark browns and blacks, and xanthan for yellows. The lavender mutation suppresses the erythrin and reduces the melanin. With the red and black drained away, you are left with a cool-toned purple-gray base. Because this trait is recessive, a snake must inherit two copies of the mutant gene to express the color visually. A snake with only one copy is called a het, a hidden carrier that is indistinguishable from a wild-type corn snake. When two of those hidden carriers breed, their DNA combines to create a precise 25% probability, a 1 in 4 chance, of producing an offspring that inherits both mutant copies. That sudden appearance of a premium morph from an ordinary pairing is a flawless display of mathematical genetics crossing generational lines. But that still leaves us with the furious buyer staring at a brown hatchling. The answer there lies in a process called ontogenetic color change. While some reptile morphs display their final colors from day one, lavender corn snakes develop theirs over months. They must literally discard their muddy juvenile skin layers over a long period of time to reveal the hidden pigmentation underneath. Between 6 and 18 months of age, every shed cycle accelerates this physical shift. The background scales transition towards silver-purple, and the dull markings deepen into a dusty mauve. The adult markers only emerge later. The snake eventually develops thin violet-gray borders around its pattern, and the reduced melanin in its eyes turns the iris a deep burgundy. A lavender hatchling is a living genetic blueprint, waiting for a specific chronological window to execute its final design. However, possessing the right DNA and waiting 18 months is still insufficient if the environmental conditions are wrong. This heat map shows the thermal gradient required to power a corn snake's metabolism. It needs a warm zone resting at 85 degrees Fahrenheit and a cooler 75-degree ambient zone. Without precise temperature control, the animal's metabolism slows. This can disrupt the shed cycle and stall the ontogenetic color change, leaving the snake looking dull. Additionally, under warm 2700 Kelvin incandescent light, purple tones look remarkably brown. Only under daylight spectrum LEDs do violet hues fully manifest. The discrepancy between the buyer's expectations and the animal in the box was a result of the specific, often counterintuitive nature of reptile biology and time. Patience and precise husbandry are what allow the lavender's full aesthetic potential to emerge. To explore advanced lavender combos, check current market prices, verify genetic documentation,